many of you printed out the POGL activity worksheet and brought it with you? Dang it, people. That's right, I made 50 copies because I was wondering. So here's what we're going to do. I would imagine that we're going to get to this activity by the end of class, probably. I don't know, we'll see. And so I don't want to take time at the end of class to pass these out because I think we're going to be tight for time. And so I'm going to pass these around, and if you need one, take one so that you have it and we can just start into that activity hopefully at the end of class. I think what I'm going to do is actually make that activity your second homework assignment. So a little bit about POGL. It's all the rage right now in pedagogy. It's something that I've never tried, so you guys are my guinea pigs. And so normally I stand here and I lecture and I talk about everything that you need to know, and you guys right now, and that's kind of how I usually do my classes. But I'm always up for experimentation and trying new things and getting feedback from you guys on how things are working or not working. And so we're going to try this activity, and the idea behind POGL is it is kind of self-guided learning, if you will. So I'm going to stop lecturing, and you guys are going to get in a group, and you guys are going to work through this worksheet. And hopefully by doing so, you're going to learn what I would have lectured about for epithelial tissues. So we'll see. And I want your feedback on it, and I'll ask for it formally probably the next time that we meet, maybe through a quiz, and then you telling me how you felt about this activity. But it is a group activity. We'll use it as your second homework assignment, so I'll have it due next Tuesday when you come to class. And there's pretty specific instructions on the beginning of that page that kind of shows what we're going to do as far as that POGL activity. So hopefully that's something that we have time to kind of mostly do in class today. And if not, it's something that you can take home and continue to work on and bring back to class on Tuesday for your second homework assignment, essentially. Lab exams next week, so just a reminder about that. There will be an open lab Saturday morning here. And then I will do my open lab, and I think Corey will be doing his open lab on Monday morning. But after that, we'll be setting up the exams in the lab rooms, and so you won't have an opportunity to be able to come in. So that's kind of your last shot Saturday and Monday morning if you are wanting to get into the labs and do a little bit of extra studying for that open lab or for your exam. The only other thing that I want to say is I sent out an email yesterday, and in that email I had some information about an extra credit opportunity. So many of you have probably heard that the bodies exhibit is coming to Boise, which I am completely ecstatic about. And if I had more time, I would love to volunteer and like do tours or something there. But if you go to that, and bring me your ticket stub, write a little synopsis about what you learned, what maybe the most interesting thing was, and I will give you 8 points of extra credit. That's about 1% of your grade. The other thing, going to a bodies exhibit is expensive. It's like $18 a ticket. If that's something that you don't want to do or you can't do, I always, every semester, give the opportunity for students to earn extra credit through community service. So that could be anything. Okay, maybe you volunteer regularly at your church, or you volunteer at a food bank, or you just go and help out one day a week at your kids' school. Specific instructions are in that email that I sent out. I also gave some things that you could do if you don't have something that you're doing regularly, like donating blood to the Red Cross, or Lake Lowell actually has a Habitat Restoration Day coming up, and the information for that is there. So if you don't want to do the bodies exhibit for whatever reason, then you can still earn that 8 points, which is 1% of your grade, by doing that community service. Yeah. So
is that's an either-or type deal?
It's an either-or, yeah. So I'm actually, by departmental policy, limited on the amount of extra credit that I can give for my class. And so I can't just, oh, okay, great, you did this, here's 8 points, you did that, here's 8 points. So it's an either-or.
Where's
thing.
that exhibit at?
It's at the Discovery Center, and I think it's opening next week.
The 29th.
Yeah, the 29th. So I'm super excited. I've been like traveling all over the country trying to hit it, and I'm always there like right before it comes or right after it leaves.
It'll be open for how long?
It's going to be open until March.
March, okay.
Yeah, so I'm super, super excited. And maybe it's not something that you want to do like immediately. Maybe it's something you want to do when you're in 228 next semester and you've got like all this background. So maybe the community service this semester and that next semester, or go both, like, whatever. Any other questions before we get started?
Yeah, I understand that. It's really cool.
Yeah, that's what I've heard. Go ahead. the bodies exhibit has bodies, real human bodies, that have been plasticized, meaning that they have been preserved by filling them with plastic essentially, so that you can see all the structures and all the details, and they take them apart. So they are real human bodies. It's basically a cadaver exhibit. And that is something to be excited about. Everybody should see the look on his face. It's great. This is very exciting. I am pumped about it.
Slice bodies of real fish.
I've looked at many books about it, and it's really cool how you can see all the different body systems and stuff like that.
Yeah, I'm pumped. I'm totally excited. I'll probably be there on the 29th. And I'll probably spend 12 hours there. So if anybody wants to come that day and wants to tour. Yeah.
I went to one in Seattle.
Well, that's the thing. I'm okay with that. Pure blood, no problem.
So it's exciting. All right, guys, let's bring it back together. So we finished up the last time we met talking a little bit about the difference between active and passive transport, and then we looked at some specific examples of passive transport. So this is a situation where substances are crossing the cell membrane, but they're being driven to do so by natural processes. Basically, the kicker is there's no energy being expended to move these substances across the cell membrane in simple diffusion, in facilitated diffusion, in osmosis. They're just moving from their area of higher concentration to their area of lower concentration because of collisions that are happening in that area of higher concentration. And so we finished up kind of talking about passive transport and looking at some examples of it on Tuesday, and that leaves us to talk about active transport. So you may remember from when we started talking about these transport processes on Tuesday that with active transport we've got a situation where substances are moving from their area of lower concentration to their area of higher concentration. And that, of course, goes against what would naturally happen, because where are we going to be having the most collisions to push things away, to disperse things, when we've got a concentration gradient? But in the area of higher concentration, right? So we're going to see with this particular situation that we have to expend energy, because they're not going to naturally disperse from an area of lower concentration to an area of higher. We're going to have to expend energy to actually pump them from that area of lower concentration to their area of higher concentration. So you may be wondering, well, why the heck would the body expend energy, okay, in the form of ATP? Why would we use our ATP reserves to do this, okay, to constantly pump these substances against their concentration gradient or from their area of lower concentration to their area of higher concentration? Well, I'm going to mention a couple of reasons here. Basically, all the cells of our bodies are doing this at all times. They're expending energy to create these concentration gradients and maintain these concentration gradients, because muscle contraction cannot occur without a concentration gradient. Nerve impulse conduction, which is the electrical signals that are generated by the brain, by the spinal cord, they don't occur as well unless there's a concentration gradient. So there's some very important reasons that we see basically all the cells of the body at all times expending the energy necessary, using ATP essentially to constantly pump substances against their concentration gradient, or from an area of lower concentration to an area of higher concentration. It needs to maintain those concentration gradients. And it's active transport that allows this to actually occur. So I want to kind of remind you of what active transport is. We had kind of an abstract definition on Tuesday, and here we're going to get into more visual representation for those of you that are a little bit more visual. And again, the idea with active transport is we're doing the opposite from what we see with passive transport. And we've got substances that are moving from their area of lower concentration to their area of higher concentration. So again, if you look at this, you can think of this as a cell. We've got some molecule that's found both inside the cell and outside the cell. It's at a higher concentration inside, a lower concentration outside, and active transport is moving that substance into the cell. And of course, that's going to require energy, because we're not going to have very many collisions that are happening out here because of our low density to push things into the cell. We're going to be having much more collisions occurring that are actually going to work to push things out of the cell. Now, if you think about what's going to be happening with active transport, we've got this situation, we're expending energy, we're constantly pumping these substances into the cell, okay, in this case, against their concentration gradient from an area of lower concentration to an area of higher concentration. And active transport is going to set up and maintain this concentration gradient. It's going to prevent this from running to equilibrium because we're constantly pumping stuff in, and by constantly pumping stuff in, we're keeping this area in here highly concentrated with this molecule, and we're keeping a lower concentration out here, okay? So we've set this up with active transport, we've used energy to do so. Anytime we have a concentration gradient, what's going to then start to follow? Passive transport, okay? So we're going to be having more collisions in here. These things are going to be bumping into each other, spreading out into opposite directions. And so we're expending energy to constantly pump them in, and we've got to keep doing that over and over and over, because what passive transport is going to lead to is them diffusing back out. So it diffuses out, we have to expend energy to pump it back in to keep this concentration within here high.
So it would be like putting water into a boat with a hole in it, let's say.
Pretty much, yes.
Okay.
And so you have to work hard, right, and expend a lot of energy to keep the boat filled with water when it's constantly also running back out. Perfect analogy there. So we've got this continuous pumping and this continuous expenditure of ATP, okay, the fuel for our body, to allow us to maintain this concentration gradient, essentially, is the idea behind active transport. So what I want to do is talk about a couple of specific examples of active transport that actually happen within the body. So we're going to look at first what's known as primary active transport, and then we're going to talk about what's known as secondary active transport, and then we're going to talk about endocytosis, exocytosis, and transcytosis, which are all, again, active transport processes where we are expending energy in the form of ATP to pump something in one direction or another, essentially across the cell membrane. So we'll start out with primary active transport and secondary active transport. And I want to say a couple of things about these two processes before we start getting into the specifics. First of all, I'll say that primary active transport directly uses energy from ATP to pump molecules. Secondary active transport does not. So secondary active transport indirectly uses ATP, or indirectly uses the energy from ATP to move molecules across the cell membrane. So that's kind of the basic difference between those two. And with that having been said, what I want to do now is show you the examples of why this is actually the case. And we are going to start out with talking about primary active transport. So again, I've got a diagram of a cell here, and we've got a situation that's occurring basically in all the cells of the body at all times, where we're using what are known as sodium-potassium pumps. These are little pumps that are embedded in the cell membrane itself. All cells of the body are going to have thousands of them, essentially. And we're using these sodium-potassium pumps to pump potassium into the cell and pump sodium out of the cell. So we establish and we maintain this concentration gradient in all the cells of our body. And the reason that we do so is we need a certain concentration of potassium inside the cell, and we need a certain concentration of sodium outside the cell for muscle contraction to work, and also for nerves to be able to send electrical signals, essentially. But a consequence of this situation where we're constantly pumping potassium in and we're constantly pumping sodium out is, of course, we're establishing and we're maintaining a concentration gradient. And what we have going on as far as concentration gradients in all the cells of the body is a situation where sodium levels are higher outside of the cell because we're constantly using energy to pump sodium out, And yeah, it diffuses back in, but then we pump it back out, okay, to keep that concentration gradient maintained. And we've also got a situation in basically all the cells of the body where potassium levels are higher inside the cell, okay, because we're actively, constantly expending energy to pump potassium in. And yes, it diffuses out, okay, because we have a higher concentration inside, but then we expend that energy to pump it back in so that we can keep potassium levels inside the cell higher than potassium levels outside the cell. And again, we need to do that because muscle contraction is reliant, okay, on that difference in concentration gradients on the two sides of the membrane, and nerve impulse conduction is as well. So if your cells ever run to equilibrium where you have equal concentrations of sodium on both sides of the membrane, equal concentrations of potassium on both sides of the membrane, your muscles cannot contract, your nerves cannot fire, and basically you're dead if you hit equilibrium in those cells with sodium and potassium. So this is a great example of active transport that's going on in basically all of the cells of your body at all the time to maintain these concentration gradients. And each time that we pump potassium in and we pump sodium out, we have to break down a molecule of ATP, and that uses a significant amount of our energy stores, of our ATP in the body, is just pumping that sodium out all the time and pumping that potassium back in, as ridiculous as it sounds. Yes?
So the potassium, is it the sodium potassium pumps, is it both? Is it not just sodium, or is it not just potassium?
No, they are coupled together. So as it's moving potassium in, the same pump is working at the same time to move sodium out.
Oh, okay.
Yeah, they're coupled together. They're not separate pumps, and of course it's more efficient that way, because if you can do an exchange and only use one ATP, that's better than using an ATP to move a sodium out and an ATP to move a potassium in. Anything else that I could explain about this better before we move on?
Sorry, is the potassium, is it just like pulling from whatever's outside, or and hoping that it gets potassium, or is it actually targeting potassium?
It's targeting potassium. So those sodium potassium pumps, the channels that those ions move through are specific for sodium. They only transport sodium, and they are shaped such that they can only transport sodium out of the cell, and they are shaped such that they can only move potassium into the cell. So that it's like a locking key, and they only accept those two ions. Anything else? Yes?
What muscles are these, is this occurring in all muscles?
All of them. Yeah. So for you to be able to contract your muscles, that concentration gradient has to be maintained before that contraction can ever occur. And when people start to get crampy, very, very often it's because they are getting a problem with levels of ions on the two sides of the membrane. So they're not where they should be.
Muscle as well as all the rest.
Smooth muscle, cardiac muscle, skeletal muscle, yes. They all are using levels of sodium and potassium, as well as your brain. So most of the things in the body are working off of sodium and potassium concentration on the two sides of the membrane.
That's why when you have Charlie horses,
Exactly. Bananas have a lot of potassium, and so when you start getting those cramps, it's usually an ion deficiency, causing a discrepancy in what the concentration should be for sodium or potassium on the two sides of the membrane. Yeah?
Earlier you said this happens in all cells of the body all the time. Is it just all muscle cells and nerve cells, or all cells?
It's happening in, so it's not every single cell in the body. Some cells of the body aren't even living, but most of the cells of the body this is happening in it. It's not just muscle cells and nerve cells. All right, so that is primary active transport, and again, each time that we pump sodium out and we pump potassium in, we're going to be using a molecule of ATP, the gas of the body essentially, to be able to do that. Secondary active transport is a little bit more complicated. It's actually a 3-step process. And so secondary active transport builds on primary active transport. Primary active transport has to happen first for secondary transport to ever even occur. And so what we're going to do is talk about in step 1 and step 2 the necessary precursors, the things that have to happen first for us to be able to have secondary active transport or co-transport occurring. So I want to, the weather is terrible and the air quality is bad. I'm having a hard time even breathing. I want to bring your attention to this key up here. If you look at what's going on, arrows in black are indicating primary active transport. Arrows in red are indicating simple diffusion. The only place in this whole series of steps where we actually have secondary active transport occurring is right here with the green arrow. So I don't want you to think that this whole process is secondary active transport. This is primary active transport, which sets up a situation where secondary active transport can occur, essentially, is the idea here. And so now what I want to do with that having been established is go ahead and walk through each of these things that need to occur for secondary active transport to be a possibility in the body. The first step is, as I said previously, you've got to have primary active transport occurring. And so we've talked about how most of the cells of the body all the time are constantly pumping potassium in, they're constantly pumping sodium out, and they're expending energy to keep doing this over and over and over and over again. And so we end up with, as we talked about with the last slide, a situation where, because we're always pumping that potassium in, we've got a higher concentration of potassium on the inside. Because we're always pumping sodium out, we've got a higher concentration of sodium on the outside, okay, that we're maintaining. We're basically maintaining these concentration gradients. Well, we also talked about previously that when you have concentration gradients, okay, what's going to happen next naturally is simple diffusion. You're going to be having more collisions of potassium inside the cell, which is going to tend to drive potassium back out. You're going to be having more collisions of sodium outside the cell, which is going to tend to drive it back in. And so that's what we're seeing here in this third step. We've got a higher concentration of sodium outside the cell because we're constantly pumping it out. Because it's at a higher concentration outside the cell than in, sodium, through simple diffusion, is going to start moving back into the cell. Okay, just because those collisions that it's having outside the cell is pushing it away from that area where it's highly concentrated. One of the things that I said when we first started talking about passive transport and simple diffusion is when you have an ion or a molecule that's moving down its concentration gradient from its area of higher concentration to its area of lower concentration, there is some kinetic energy that is associated with that movement. And we can use that kinetic energy to move other things into the cell. And so what we actually see happening as sodium diffuses back in is we use some of the energy from sodium moving naturally because of the collisions to actually bring molecules of glucose into the cell as well, so that we can use that sugar for energy inside of the cell. So what I want you to kind of take from all of this, I guess, is that our bodies are as efficient as possible. Okay? Over here, we've got the movement of sodium and potassium being coupled together so that we're using one ATP to pump both of those. Over here, we're recapturing that energy that we use to set up this concentration gradient to allow us to move something else into the cell without now having to use another ATP to pump it. It's coming in through the energy being released by simple diffusion.
So that glucose is coming through from integral proteins and active transport.
So it is big, and it would have to.
It's not going through the phospholipid.
No, it's not. It's way too big. Anything else?
So is this like a circle of this is what happens? So it like does that and then it starts back again, or is that?
Right, so this is circling through all the time. So we're constantly maintaining this, which causes sodium to diffuse back in. It's going to bring glucose with it without us having to expend any more energy. And then as sodium's diffusing back in, we pump it back out. It diffuses back in and it brings glucose with us. So this process is cycling all the time, and basically what we're doing is we're recapturing this original energy that was expended over here to move those two things so that we can now like move something else along with it, essentially.
So we want to be right there, though. Like that's what our whole goal is.
So we want to maintain that concentration gradient so that muscle contraction can occur, but also so that we can bring in other things into the cell as we have that diffusion occurring.
So we have to have the glucose to help make energy?
Yes.
To maintain our equilibrium.
Right.
Right?
So yes, I'd never thought about that, but you're right. The reason we're bringing glucose in is so that we can make ATP, so that we can maintain that concentration gradient.
And that's why when people get low sugar levels, they're like, you know.
When you get low sugar levels, the first thing that's affected is the nervous system, because most other systems of the body can switch over to breaking down fat and converting it to glucose and then running the reaction. The brain does not do that. And so if somebody starts to get hypoglycemic, they get shaky, they start to get confused and say really weird things because the nervous system isn't functioning properly. It's not switching over to fat, and it needs that constant supply of glucose. Anything else? All right. So we've talked about some kind of complicated things. The next 3 types of active transport aren't really so bad, and I'll just mention them quickly. The first of these is a process which is known as endocytosis. So the idea behind endocytosis is you've got some very large thing that's outside the cell, maybe a protein, maybe a large molecule that's too big to cross through the phospholipids. It can't fit through a channel, it can't fit through the protein or through a transport protein. But it's something that the cell wants to take into its body to use for whatever reason. So if you look at this picture here, we've got a cell. Here's our cell membrane in the dark brown color. Here's some large particle outside the cell that the cell wants to take inside of its cell membrane. And you can see how this process of endocytosis occurs. Essentially, the cell membrane is starting to invaginate around that particle. What's eventually going to happen as that cell membrane evacuates around it is, remember, these cell membranes are not rigid things. They are like soap bubbles, okay, so they can break and reform. And what's happening essentially here is the cell membrane is going to pinch off, okay? It's going to close back up, and when it does so, this particle that was previously outside of the cell is now going to be inside of the cell, surrounded by a part of the plasma membrane or part of that phospholipid bilayer, and it's entered the cell without ever having to cross the cell membrane. So it didn't go through the phospholipids, it didn't go through a protein or a channel or anything like that. And then what will happen is we've got little digestive enzyme organelles that are inside the cell that will come and digest the cell membrane, digest the phospholipid bilayer, break it down, and we will then have that particle that was outside the cell now inside the cell for the cell to use as it would like. So that's kind of the basic idea of endocytosis. Also an active transport process because it requires energy in the form of ATP to break and reform the cell membrane essentially when we have this going on. Exocytosis is basically the reverse process of endocytosis. So if you look at this picture down here for a moment, focus on this. We've got a cell, and you can see that it's actually releasing some things that were produced in the cell to its external environment. And cells are doing this all the time. They're little factories. They produce proteins and lipids and other types of things that the body needs, and then release them to their external environment where the body can actually use them. So that gives you kind of a nice overview of this process of exocytosis. I want to talk a little bit more about the specific steps that are involved. So if you focus on this part of the picture here, we've got that phospholipid bilayer, okay, that makes up the plasma membrane or the cell membrane. This is the area inside the cell. These little green dots are representing something. We'll say it's a protein, okay, some protein that was produced in the cell that needs to be released to the outside of the cell. And one of the things that happens, and we'll talk about this a little bit later on, is when a cell actually produces something, before it releases it, it packages it up in these little sacs, which are known as vesicles. And vesicles are basically structures that are composed of a phospholipid bilayer. Okay? So we've got some protein in here, it's got this little sac that's surrounding it, and you can see that this sac is moving towards the cell membrane. Then what happens, you can see that illustrated here, is the phospholipid bilayer that forms this vesicle, that forms the sac around our product, actually attaches into the plasma membrane. As it does so, that releases this protein into the external environment of the cell, and now we're allowing something that was really too large to squeeze through the phospholipid bilayer, couldn't fit through a channel, couldn't fit through a transport protein. We're allowing that substance to be released from the cell without it ever actually having to cross the cell membrane itself, essentially, is the idea here. And again, an active transport process, because we're having to use energy in the form of ATP to move that substance across the cell membrane. So the last type of active transport that I want to talk about before we move on away from the cell membrane and don't worry about it anymore is a process which is known as TransIPTosis. And if you think about this term, we've talked about endocytosis, we've talked about exocytosis. TransIPTosis is a process that kind of combines endocytosis and exocytosis. So if you look at this picture here, okay, you'll notice that we have a cell barrier. We've got some tissue that's composed of cells that's making up a membrane. What the artist is actually trying to represent this as is the lining of the vaginal canal or the lining of the anal canal, okay? So this would be the anus here or the vagina. Here is the membrane, okay, that kind of lines that cavity within the body, and this would be the area inside the body itself. And this particular picture shows how HIV uses this process of TransIPTosis to actually get into the body and cause an infection. There was a paper that was published, it's been a long time now, it's been about 15 years in Nature in 1997, and of course Nature is one of the top journals in the world, if not the top journal in the world. And this particular paper, through research, had shown that HIV actually is capable of crossing cell membranes, okay, even if there isn't a break or a tear. You don't have to have a cut in the anal wall. You don't have to have a cut or a tear or a sore in the vaginal wall for HIV to actually be able to penetrate through it, because it uses this process of TransIPTosis. Okay? So if you look at what we've got going on, anal canal or vaginal canal, whatever you prefer, here are some immune system cells. We talked about the last time we met how HIV actually gets into the cells of the body by exploiting proteins, peripheral proteins, GP120, CD4, CCR5 were some of those that we talked about. So if you get exposed to HIV, because that virus is deposited through body fluids into the vaginal canal or into the anal canal, there are immune system cells in that area. And HIV within the vaginal canal or within the anal canal will get into those immune system cells. Now that's not such a bad thing there, because that's still technically outside the body. You don't have an HIV infection. But the other thing that HIV will actually do is it will interact with the cells that are lining the anal canal or lining the vaginal canal. It will have those cells pick them up through endocytosis. So it actually gets into those cells, okay, through a vesicle, travels across that cell membrane, gets released on the other side by exocytosis, and now HIV is in the body proper. And once it is in the body proper, it can now start interacting and getting into white blood cells that are in the body, and you now have an HIV infection without a sore or a cut or a scrape or something like that that we would typically think of would be necessary for that particular infection to be spread. Happens through TransIPTosis, and that was shown a very long time ago. There's one last thing. Sorry, I said that was my last thing about cell membranes, and I lied. This is my last slide about cell membranes, okay? And that is, I want you to know that the cells of the body have an electrical charge to them, okay? Or the membranes have an electrical charge to them, essentially. This electrical charge that the cells of the body have is known as the cell membrane potential. What causes this electrical charge essentially is selective permeability. So you'll remember that's the idea that cells allow some things to cross better than they allow some other things to cross. The potential for cells, the difference in voltage that we typically see is usually somewhere between 50 and 100 millivolts. In muscle cells and nerve cells, it's usually about 70, and we'll talk about that when we get into chapter 9 and we're looking at muscles, and when we get into chapter 11 and we're looking at nerves. But I've drawn a picture that hopefully will help to illustrate this for you a little bit. Here's my representation of a cell, and I mentioned that we've got this difference in charge on the two sides of the membrane, and what the specific difference is that the outside of the cell membrane is positively charged relative to the inside of the cell membrane. And so I've drawn this plus and this minus here to help you remember that particular fact. What I want to talk about a little bit is why this is the case, okay? Why the cells of the body have this charge, or this difference in charge, this membrane potential on the two sides of the membrane. And it goes back to some things that we've already been talking about. We've already established this idea that there's more sodium on the outside and more potassium on the inside, because sodium-potassium pumps are always pumping sodium out. They're always pumping potassium in. So we've got this concentration gradient that's established, that's being maintained through active transport essentially. We also talked about the fact that anytime you have a concentration gradient that's established by active transport, that you're always going to have those ions diffusing from their area of higher concentration to their area of lower concentration. So sodium's always diffusing back into the cell, potassium's always diffusing back out of the cell. But the cell membrane itself is much more permeable to potassium than it is to sodium. It's about 75 times more permeable. And because of that, we have a situation where more positive charge is leaving the cell, less positive charge is entering the cell through diffusion. So our net movement of positive charge essentially through diffusion is out of the cell. And that's why we can say the cell membrane on the outside is relatively positively charged. The cell membrane on the inside is relatively negatively charged. They're both due to the movements of positive ions, but this one is more positive. This one is less positive because less positive ion is moving into here. And so another easy way of saying that is just to say that we're positive on the outside, negative on the inside, essentially. Anything that I could explain better with regards to that?
So, when the because we have potassium in the cell and it's leaving, the potassium on the outside makes it positive, right?
It does. So potassium is a positive charge.
And the sodium that's coming in is making the inside negative.
So it's not making it negative because sodium is also a positive charge. There's just more positive charge going out than there is positive charge going in. And so we've got more positive charge on the outside and less positive charge on the inside, basically. Anything else? Yeah?
So because it is less charged, the cell is, would it make the process for the cell to grab onto things more easily because it has a little bit of a charge?
So that's related to pseudopodia, and so these charges are in millivolts. They're very, very small, which is why, you know, if you touch yourself, you don't get shocked or anything. But what it does allow for is muscle contraction and nerve impulse conduction to occur. Yeah, so it's not grasping onto things. That's done through the proteins on the outside. But more electrical activity that's happening in the body, and muscle contractions and electrical activity, and nerves firing is an electrical activity as well.
I have a question. Why is the sodium moving slower than the potassium?
Because the membrane's just not as permeable to it. So it's not as happy about sodium crossing as it is with potassium.
So it's nothing to do with size?
Has nothing to do with size. It just has to do with the membrane being selectively permeable and allowing one to cross at a higher rate than the other.
Okay, thank you.
Yes.
So how does, okay, so when the potassium potassium moves out, does it kind of just hang out around the cell?
It does. So it's just in that outer environment of the cell.
Okay.
All right. So this next part I'm going to try to go through quickly so we have time for our POGO activity. I wanted to talk just briefly about organelles. The analogy that I usually use for teaching about cells is that they are a factory, okay, and they are producing things for the body, proteins and lipids and whatnot. And as a factory, they need to have all of the same types of things that the sugar factory across the street or any other factory would have. That is, they need to have an area for production, they need to have an area for packaging their product, they need to have little forklift operators that are moving the product out of the factory. They need to have an energy source, they need to have security and maintenance and all of those types of things as well. So as we're talking about these organelles, we're going to be talking about them kind of from this factory analogy. What specifically is their role in this factory that is the cell? And I'm going to start out with the ribosomes. They are a production area of the cell factory, and what their role is to produce proteins. If you look at this artist representation of a cell here, some of the ribosomes, they're represented by these little red dots, are just floating freely in the cytosol, that jelly-like fluid. Other ribosomes are actually embedded in the endoplasmic reticulum. But they are an area of production for the cell and are working to produce proteins. You guys also learned in lab to identify the endoplasmic reticulum. So this is also kind of a production area within the cell. It produces lipids. Some of it has ribosomes embedded in it, so I guess you could say in a roundabout way that it works to produce proteins as well. You probably talked about in lab the fact that endoplasmic reticulum can be either rough or smooth. So some of it has ribosomes embedded in it. It's studded with ribosomes, and you can see that here. That would be an example of rough endoplasmic reticulum, because when you looked at it under a microscope, it would actually have that studded appearance to it because of the ribosomes embedded in it. There's also what's known as smooth endoplasmic reticulum, and you can see some examples of that over there, which does not have the ribosomes embedded within it, but again is kind of playing that same role, producing lipids. Producing lipids within the cell and acting as a production area within the cell itself. Golgi apparatus, you can see represented here in green. So this is our packaging area, and what happens after these proteins are produced on the ribosomes, produced on the rough endoplasmic reticulum, is they get shipped to the Golgi apparatus. The Golgi apparatus actually pinches off a part of its organelle wall, packages those proteins in a little vesicle, one of those sacs that we talked about previously, and then gets them ready to be exported from the cell by exocytosis. So the Golgi apparatus, again, is basically the packaging area of the cell.
Is the Golgi apparatus made of a phospholipid bilayer?
It is, yeah, because that bilayer is going to have to join with the cell membrane and become a part of the plasma membrane. So it's exactly like the plasma membrane. The other last thing that I wanted to mention with this particular slide are the mitochondria. So this is kind of our energy source, our powerhouse for the pump. This is where ATP, that fuel that our bodies run on, is actually being produced. So you can see several of these represented in these cells. Mitochondria are pretty predominant in cells. On average, cells have about 1,700 mitochondria actually within them. So this is a very simplified representation of the cell. When you look at skeletal muscle, which has to have ATP being produced all the time to power muscle contraction, there are thousands more mitochondria present, and cardiac muscle has tens of thousands of mitochondria actually present in each of the individual cells, constantly working to produce ATP so that the heart always has what it needs as far as energy to continue to contract. Lysosomes and peroxisomes are two organelles that are kind of similar in their structures. We've got a peroxisome here, a lysosome here. Both of these organelles contain digestive enzymes within them. So they are working, in the case of lysosomes, to destroy foreign invaders, to break down cell parts that have actually become worn out. So you could think of them as kind of like security and maintenance within the cell factory, if you will. And the same idea with peroxisomes. They contain enzymes specifically which break down fats, alcohol, and other molecules. Based on what peroxisomes do and the things that they break down, what tissue of the body would you expect to find them most predominantly in? The liver, okay? Because that's the organ of the body that's processing alcohol specifically. And so there are a lot of peroxisomes in that particular organ. We've talked about vesicles a little bit. Okay, you can see here the Golgi apparatus and a vesicle, a little sac, okay, a phospholipid bilayer that contains something within it that's pinched off from that Golgi apparatus that's moving to the cell membrane to allow something to be released by exocytosis. Vesicles are also produced when endocytosis occurs, okay, and we take something into the cell that's surrounded by a little portion of the plasma membrane. That's a great example as well, just little sacs that are transporting substances within the cell. All cells have a centrosome. So if you look at this diagram over here, this shows us what a centrosome is. It's basically consists of a couple of structures which are known as centrioles. So the whole thing here is a centrosome. This is a centriole. This is a centriole. And we find one of these in each of the cells of the body. And what they're actually used for is to pull chromosomes apart during cell division. So if you guys remember from your labs, okay, one of the things that starts to happen in prophase is we get production of this structure which is known as the mitotic spindle. And the mitotic spindle starts to spread and extend towards our chromosomes that are located in the center of the cell. Well, what that mitotic spindle is actually formed from are the centrioles. So you have one of the centrioles move to one side of the cell, the other centriole actually moves to the other side of the cell. These little tubes, okay, that make up the centrioles start to extend out towards the chromosomes, actually attach to them, and then retract back into the centrioles to pull those chromosomes in opposite directions within the cell itself. So to make sure that we have enough time for our POGL activity, I think I'm going to skip covering the cell cycle. This is something that you talked about, at least should have talked about, basically interphase and the stages of mitosis within your lab classes, so I won't rehash that here. That'll give us, it looks like, about 25 minutes for you guys to organize yourself into your groups. Because we're not covering this, that means that I also won't test over this on your upcoming exam, and we'll just have to go with what you talked about in your lab classes as far as knowing the cell cycle. So where did those sheets end up that I passed around? Okay. When I get back on my desk, did I pass them out?